I'm going to begin with this webinar in the root camp episode 3, integrating soil biology for plant nutrition. And I would like to talk about this wide topic, but the main idea of my dissertation is about soil health, beneficial microorganisms, and sustainable agriculture. If we have to talk about sustainable agriculture, maybe we have to talk a lot of things, but if we think as agronomists, we have to think in the soil. The soil is the most important part of our ecosystem, and we need to take care of it. In this idea, we need to have the main important thing, that is to have the soil covered with vegetation, with plants, because it's the only way that the soil can keep soil organic matter. And if you, the overview of this presentation is including the idea of sustainable agriculture and soil microorganisms. The soil microorganisms are very important because all the processes in the soil, all we need to have plants on the soil is because the soil microorganisms are working. In this idea, I have to project the part of the soil, and I'm going to talk about the rhizosphere effect and the difference with bulk soil. And also I'm going to talk about the microorganism-plant interaction, symbiotic association, and non-symbiotic associations like PGPR, or plant growth promoting rhizobacteria. And finally, I'm going to talk about the biotechnological applications and how we can do that with a very important formulations and quality control. Talking about sustainability in agroecosystem, it's important to say that we are going through two different paradigms. The paradigm of the last century, with the called Green Revolution, which is which was including a high levels of enzymes, chemical enzymes like pesticides, fertilizers, and fluid oils, and fossil fuels, and this amount of enzymes degrade the soil. So in some way, we now are going through the idea that sustainability can be recuperated, and we can have a high level of sustainability. If that idea is because we are going through a new paradigm of the bioeconomy. The bioeconomy paradigm is named also the New Green Revolution because it's including the sustainability, sustainable agriculture, and intelligent industrialization. And because of this idea, we have to say that if we increase the use of the biological information, like biological control, use of PGPR, biological nitrogen fixation, and another microorganism plant associations, we get the recuperation of the system. We can go through to the sustainable system, because sustainable systems are those which are capable of achieving high production without the at least negative effect in the environment. So with this idea, we have a curve I would like to see, but if we have high levels of chemical enzymes, the sustainability is decreasing. And if we use information and use biological combination and keep the biology of the soil, we increase sustainability. Because we are saying that we have going through the last century, the 28th century paradigm, with the use of high levels of pesticides, fertilizers, and fossil fuels, and the new paradigm of the bioeconomy, which is including sustainable agriculture and intelligent industrialization. As I had told you, if you use high levels of chemical enzymes, we have decreasing in the sustainability. And if we use more information about biological associations, plants, and microorganisms, we can get a recuperation of the sustainability. And because the sustainability systems have to have the highest production without the least negative impact for the environment. In this new green revolution, you have to have in mind that there is underground key points. These underground key points are very important because, as I told you, and maybe everybody is agree, that the soil is the most important part of this ecosystem to keep or to reach the sustainability. So how the soil organic matter is formed, and you can see there the emerging understanding of these processes. And the roots, the plant roots, are the central part of this situation. And the rhizosphere effect, which is the part of the soil which is surrounding the roots, is very important. And in there, you can find the most diverse ecosystem in the biosphere. If you go through, I can show you on the left different key points, underground points. The soil biology is the central part of the new green revolution because we can keep, because of the activity of the plants and the rhizosphere associated, because in there you can have what is called rhizodeposition. The roots exudate and excrete a lot of carbon substance which are attracting microorganisms and other soil organisms, and all the processes that we can think like the production of all micronutrients in the soil solution are related to microorganisms activity, and also soil organic matter. The degradation of organic matter is associated with the activity of plant roots, rhizosphere effect, rhizodeposition in that environment, and the activity of soil microorganisms. So with this idea, we can reduce the ecological deficit because the biocapability is not possible to change. Every ecosystem has that, the particular one, but we can reduce the ecological fingerprint. So on the next one, you can see and you can think where the microorganisms are. Okay, here we have two questions. Which is the role of soil microorganisms? The three main roles are there. Efficient use of natural resources would be impossible without microorganisms. Transformation of substance to be used for another soil organisms. All the resources available for plants and other organisms like mesofauna are providing for the transformation of substance in the soil produced by microorganisms. The composition of pollutants and toxic substance is one of the important things. The soil can depurate pollutants and toxins because of the activity of microorganisms. And where are they? Where can be found soil microorganisms? Well, the soil, as you know, is an extremely diverse system, and you have extremely diverse habitats. And the two main habitats are the rhizosphere and the bulk soil. In the rhizosphere, as I told you before, is a very dynamic environment because we have the rhizodeposition of the roots, and this is attracting microorganisms and is where most of the microorganisms in the soil are. But in the bulk soil, you have different activity, and this activity is depending of the structure of the soil. The porosity is alberging different size of microorganisms, and also you have there what we name the microenvironments with different characteristics. If you see the picture of the microaggregate of the soil, is showing that the different concentration of oxygen can produce different soil microbial communities in a very short distance. So we have microorganisms in everywhere, and in the next one you can see that the microorganisms have two principal characteristics. Could you go to the next one? Okay, these two characteristics are the hyperdensity and hyperdiversity. If you see there the numbers for gram of soil, which is less than would be in a teaspoon, you have 10 million or 100 million cells, which is impossible to obtain a complete catalog. Every method can select particular group of microorganisms in the soil, but it's impossible to have all the record in a particular sample. But also the hyperdiversity. You have a lot of cells, and also you have a lot of types of the cells. Depending on the methodology, you can have 1,000 or 50,000 types of type of microorganisms per gram of soils. And in the microphotography on the left, you can see a colony of microorganisms in a particular set of soil particles. And also in the middle, you can see a aggregate and different amounts of substance or oxygen. So that would be the exemplification of microenvironments with different soil microbial communities. Continuing with all this basic knowledge, we can go through the use of microorganism-plant interactions. The rhizosphere and the root extending phenotype of the plant is a relative new concept, which would be very important that the breeders, plant breeders, have in mind. Because if you can see in that picture that the bottom of the picture is the root, and you can see the root hairs, and also the hypha of the fungi going around, and a lot of small particles which are bacteria and similar kind of cells. So this environment is very competitive. There are a lot of activities, and the plant is having some kind of protection with the good ones are the beneficial group, and is the biggest. But also we can find, as you know, the bad one, the microorganisms which cause plant diseases, and also you have the ugly ones, which one are opportunistic human and animal pathogens. When we make an analysis of the expectative to use these microorganisms as biotechnological applications, we know that the good ones, the beneficial, are the more abundant. But we have to check every time which one is the microorganisms we isolate. And it's not recommended to use the microorganisms which can have a human which are opportunistic human pathogens. So it's very important that we have this in mind. The plant is attracting microorganisms because the microorganisms help to the plant to cope with different kind of problems, and there is a phenotype of the plant associated with the rhizosphere microorganisms. And if you focus on the beneficial microorganisms, we can say that the mechanisms of plant growth promotion by microorganisms can be two groups: direct plant growth promotion and indirect plant growth promotion. On the first one, the direct plant growth promotion, you can see all the activity of the microorganisms which can be used as biofertilizers. By example, the microorganisms which have the possibility to fix nitrogen from the atmosphere, the solubilization of phosphorus or potassium, the production of siderol, which are particular substance which can chelate the iron and are very important for the nutrition of the plant, because the plant sometimes is not reaching the iron in the soil, and the microorganisms have more efficient captation of this micronutrient. So also in the direct plant growth promotion mechanism, you can find the phytohormone productions like indole acetic acid, are cytokinase and gibberellin production. On the right part of this plate, you can find some of the indirect plant growth promotion mechanisms, like production of antibiotics, the hydrolytic enzymes, and also siderophores are here, because the siderophore molecules most of the time are acting as antibiotics, and they prevent or reduce the activity of certain pathogens. And also when the plant detects some problems in the rhizosphere or also in the aerial part, the microorganisms help the plant to induce systemic resistance, and also the production of exopolysaccharides, which are protecting the microorganism itself and also prevent or help to maintain the structure of the soil. So all this combination, all these mechanisms could be important just per se, or could be combined among them. If we think in microorganism-plant interactions, we have the possibility to manipulate the rhizosphere microbiome, so the community of microorganisms which are there. And for that, we have two types of strategies: plant strategies and microbial strategies. In the center of this figure, you can find that different mechanisms we can achieve. By example, improve access to the soil nutrients, reduce disease incidence and severity, and increase stress tolerance and improve rhizosphere colonization. If we want to reach one of those, we can combine plant strategies with microbial strategies. And you can find there the groups for this manipulation. If we want to add particular microorganism, you have to select particular optimal combination between the microorganism and the plant. But also, as I probably can show you later, the plant genotypes are interacted with the association with microorganisms. So the plant strategy could be different. They can combine, and also they can combine among the plant strategy and also with microbial strategy to get the focus of the main microorganism plant interaction we want to to get. So if we go to the next one with this general information, we can begin to particular microorganism-plant interactions, like mycorrhiza. Mycorrhiza are mutualistic symbiosis. So the plant and the fungus, in this case, because mycorrhiza is the association in certain particular fungus and the root of the plant. In this plate, you can see on the left what is happening when the root is not having also root hairs. The exploration area of the root is very small. The root hair increase this area of exploration in the soil, but if the root is connected with mycorrhiza fungi, the exploration is increasing significantly. And in the center, you have a figure that is analyzing. When we talk about rhizosphere, we're talking about the soil surrounding the roots. But if we have a plant with mycorrhiza associated, the exploration is going bigger. And in this case, we talk about mycorrhizosphere, because the structures of the fungus is increasing the area of exploration of the plant. And on the top of this plate, you can see that in the center, you have the microorganisms rule, microorganism wheel, because we say that the microorganism all the time is moving the micronutrients from the soil. In this case, we are talking about the phosphorus, because the phosphorus is a sedentary element, and it's necessary that the movement of the microorganism rule move very fast to get phosphorus in the soil solution to available for the plant. So the mycorrhiza is playing a very important role in this availability of phosphorus for the plant. In the next one, I can show you also for the mycorrhiza the importance of native mycorrhiza because a particular type of mycorrhiza. Mycorrhiza, as I told you, is an association between a fungus and a root plant, but there are several types of mycorrhiza. We are talking for this one and the next one probably slides about the native mycorrhiza. The native mycorrhiza are the fungus which can select the plant which are going to colonize, and they improve the rhizosphere and also provoke different kind of ecological services for the plant. Mycorrhiza are indicator of root soil health, and in this slide particularly you can see on the table that two different crops, in this case are cover crops which are using to keep soil organic matter in different kind of agriculture systems. They have different root colonization by fungus in the soil. They are native, so we don't added any fungal inoculation in this case. And you can see different structures there, arbuscules, vesicles, and spore, and they are structures of the fungus which are connected with the plant. You can see in the figure on the left. And this is very important because the plant is having interactions in the arbuscles. The fungus make reserve substance in the vesicles, and the spores are the way that the fungus, or all general, in general, the fungi in the soil stay for longer. So in the bar figure, you can see that we can manipulate this when we use a particular agriculture practice like inoculation with some PGPR. You can see that if we talk about, by example, the inoculation with a PGPR like Azospirillum argentinense, which is a rhizosphere bacteria. I'm going to talk more about this bacterial in the following slides. But in here, I would like to show you that on the left, you have the amount of arbicules, and in the right, the percentage of vesicles, and different genotypes of wheat are associated in different way. So and also respond in a different way to the inoculation. So we have a potential to manipulate these associations using different kind of different genotypes of the plant and also the inoculation response. I can summarize here the ecosystem services of arbuscular mycorrhiza, which is the most abundant mycorrhiza because 97% of the plants are holding this kind of fungus in their roots. So you can see there that the mycorrhiza have a lot of benefits for the plant and also for the soil, because on the left and the bottom part you can see a figure. This is illustrating the production of substance like proteins excreted by the fungus that can help to stabilize the soil structure. So they help to keep the soil particles together, and they help the soil structure, which is very important to the growth of the plants, and also improve the plant growth and have a lot of benefits to reduce the amount of certain fertilizers, by example, phosphorus fertilizer, and the regulation and the increase of the plant resistance to different stressors. We are going to begin with trichoderma because the trichoderma is a fungus too that can have a symbiotic and non-symbiotic association with plants. They have a lot of benefits. They're a generous microorganisms because they have a high diversity. There are more than 400 species. Most of them are beneficial for the ecosystem and the plants, but some of them could be opportunistic pathogens. So it's very important to check when we use this kind of microorganisms and use the beneficial ones. But you can see on this plate that we have a possibility to increase plant growth and also soil health, and this microorganism can use also as a biocontrol agent for different kind of diseases. And it's very important to know that trichoderma could help the plant in different ways. Could be possible that the plant could be colonized in the roots by these microorganisms, but also could be no physical contact, and also the plant could be have beneficial effects because the trichoderma can produce different kind of substance, could help the plant like different phytoregulators, and this is very important to know. And these mechanisms combine, and it's very interesting to have this possibility to use this beneficial microorganism. On the next one, we are going to talk about other symbiotic associations, the associations between plant and the diazotroph. The autotrophs are microorganisms which can fix nitrogen from the atmosphere. So the possibility to use the huge amount of nitrogen in the atmosphere, 78% of nitrogen is in the atmosphere, is a good strategy to get agriculture sustainability. So if we include plants which can associate with this type of microorganisms, like legumes, which are associated with the bacteria of the genera, in general, Rhizobium, is a good strategy. On the top, you can see two different associations with legume plants. On the left, you see the nodules of alfalfa and the microorganism, the Rhizobio, which is associated with this plant. It's a sign of Rhizobio. The association of this type of association is very important. Is the most important, the azeotrophic association. And could you keep? Okay, thank you. And it's very, very important to know that you have forage plant and herbaceous plant, and also have crops like soybean, and also tree plants having this association. On the top, on the right figures, you can see a soybean root with nodules. The nodules are the particular structure which is generated by the bacteria with association with the plant. And inside of this structure, you have the bacteria, which is differentiate to fix nitrogen from the atmosphere. But I'm going to talk more about this association, but in here I go to show you that there are other plant diazotrophic association, like actinobacteria with the genus Branchia, which is very important in shrubs and trees. And you can see there on the left the type of nodule which is found in the roots of these plants associated with Branchia. And also in several crops like rice with a line of water on the soil, the cyanobacteria and the aquatic fern Azolla can be a very good association, because the Anabaena and nostoc cyanobacteria can fix nitrogen in cells, like you can see in the photos on the center, that there is a particular cell that can fix nitrogen in these systems. So the plant can benefit from all this nitrogen, and it can be reduced the amount of nitrogen fertilizers. But also I can point out that different plants, by example, sugarcane, sugar sorghum, and sweet potato, have inside endophytic bacteria which can fix nitrogen in very huge amounts. These bacteria are endophytic, evolve with the plant, and is reducing the amount of nitrogen which can be needed in these crops, and also have another benefits, because they can help the plant to reduce the incidence of different disease. So going through to the next one, I continue talking about the association between Rhizobia and soybean. And in the first part, you can see the figure on the left that on the y-axis, you can see the nitrogen which was fixed by the interaction between the bacteria, is the bacteria which is fixing nitrogen in association with the plant. And in the x-axis, you can see the amount of nitrogen added as a fertilizer. And this association is very strict with the amount of available nitrogen. You can see there that with 15 kilograms of nitrogen added as a fertilizer, the nitrogen which was fixed was very reduced. So this is happening because the plant have a regulation, because the nitrogen fixation is a very high energy requirement process, and the absorption from the soil is not is cheapest for the plant. So we need to have in mind, if we want to get efficient association with this type of bacteria to get nitrogen from the atmosphere instead of the soil, we have to manipulate or manage, apply agricultural management that could help this association. And on the right, you can see two roots, and this is an indicator of good and bad inoculation processes. When we make the inoculation with a good inoculant, you have the nodules, the structures I told you that are important in this association, because in there is occurring the nitrogen fixation, should be in the neck of the plant. The nodule should be in the neck of the plant. So the root on the right is very good nodulation, and on the left, maybe if you have rhizobia naturalized in the soil, the inoculation, the roots have nodules in secondary roots. So most of the time, these nodules are not efficient. So the efficiency can be checked if you look inside the nodules. You can see the red color, because the plant is receiving the nitrogen which is fixed by the bacteria, and a particular protein named leg hemoglobin is transporting the oxygen. So because it's having an iron structure, the color is red. So if we have nodules with red color, when we cut them, we are seeing the nitrogen fixation in action. I'm going to begin now with the possibility to get biological nitrogen fixation with cereal crops. In the table in this plate, you can see that wheat, maize, and rice can get nitrogen fixation. And in the second column, you can see the amount of fixed nitrogen when the soil and the plant didn't receive any inoculation. But if we add inoculation, if we include the application, in this case on the seeds of this bacteria, Azospirillum, you can increase in maize and rice the amount of nitrogen which is fixed by the system. So we have the opportunity to combine certain microorganisms like Azospirillum, which is a nitrogen-fixing bacteria, which is living in the rhizosphere and also inside the roots sometimes, to get nitrogen from the atmosphere and reduce the amount of nitrogen fertilizers. And in the pictures, you can see how Azospirillum looks like, and when you see on the microscope, and also because this microorganism can fix nitrogen without the plant, we can check in vitro the activity of this bacteria, and you can see there a graph with the growth of this bacteria without any nitrogen. So the bacteria can grow there, fixing nitrogen. So this is a very good tool to select this kind of microorganism. Well, going to the next one, I can show you the importance of this Azospirillum plant interaction. Particularly, I'm showing here the association with wheat. And in the center of the plate, you can see the figure showing different plant genotypes, varieties, which were inoculated with an experimental inoculant, and all of them increase grain yield in different amounts. So it's very important to know the effect of the plant in regulating the activity of the bacteria, and we have the tool to manipulate this information, and also to manipulate what kind of crops we want in the agriculture systems. In the bottom, you can see a line with the amount of or percentage of nitrogen fixation of this, each one of these genotypes in combination with this bacteria. And on the left, you can see that this combination can increase also aerial biomass, and in the same amount, in some way, with the amount of urea, or in this case 50 kilograms per hectare, and we can actually reduce the amount of nitrogen fertilizer. This air biomass is going to stay on the soil after we harvest the grain. So it's very important to two important things here. We have an increase of material to deposit on the soil, and also we reduce the amount of fertilizer if you use these bacterial plant associations. Okay, on the next one, you can see here the interactions between native arbuscular mycorrhiza and Azospirillum, in this case argentinense. This bacteria is very important. It's used in a lot of commercial inoculants, and is having a lot of beneficial for the plant. And in here I show you that this bacteria could help the plant to reduce the effect of the water stress. If we combine mycorrhiza with Azospirillum, we get the best possibility to help the plant to cope with the water stress. You can see that in the figure on the right, and the combination of mycorrhiza and Azospirillum is the best option to help the plant to cope with the water stress. Okay, on the next one, I show you here a particular mechanism which is very important in sometimes, and it's also, it was discovered for the Pseudomonas rhizobacteria. And here, if you see in the picture, the cell, the bacterial cell, is the yellow one, and you can see the substance there, the ACC, which is a precursor in the plant, the cell in green, and it's the precursor of ethylene. And ethylene is the stress hormone. So if the bacteria is associated with this plant cell, can get the ACC, the precursor of the ethylene, and degrade inside the bacterial cell. And in this way, we reduce the amount of ethylene. We get the plant cell healthy for more time, and is a good mechanism, which is, if you see in the cell bacterial excuse me, in the plant cell, that this mechanism is associated with the synthesis of another phytoregulator, the indole acetic acid. So we can check if the beneficial microorganism has this mechanism, and we did that for different strains, and you can see that in the table. Different strains of the Azospirillum have different indolacetic production and also ACC deaminase activity. This enzyme is inside the bacterial cell, but if the bacterial cell is associated with the plant cell, could help the plant to reduce the amount of ethylene. Okay, if we go to the next one, I'm going to talk now about the interactions between plants and Pseudomonas fluorescens. In this case, for the mechanism of the production of a particular phytohormone, the cytokinase. And on the left, you can see that different strains could grow similarly. They can produce different productions of cytokinase. And a particular strain in the table on the center, you can see that the G2018 wall type is producing growth increase because it's increasing shoot biomass and also the roots biomass. So it's the cytokinin production by the bacteria is a direct mechanism of plant growth promotion. But also, recently, we discovered in some way that this mechanism could be a way to produce biocontrol, because the cytokinase could be producing different levels of the reduction of the activity of a particular pathogen like Pseudomonas syringae, which producing a spot on the plant. This analysis was made with the model plant, the Arabidopsis thaliana, and you can see in the picture the bottom on the left that this strain reduced the symptoms of the ill, the disease in the plant. The strains have different performance, and you can choose which one is the best and could help you to increase plant growth and also to have a role in biocontrol in the plant. I show you that if we use this bacteria, in this case is for wheat, we can increase native mycorrhiza in the roots of these plants. And we can increase this with Pseudomonas fluorescens, and you can see there that could be observed in tillering and also grain filling. So you increase the colonization of mycorrhiza and also the amount of arbucles, which you can see on the left, the figure on the bottom, is one arbuscule in the cell, in the plant cell. This structure are not making any damage to the plant, to the cell plant, and in there is where occurring the interchange. The plant is giving photosynthase coming from the photosynthesis, and the fungus give water and micronutrients to the plant. So this is a very important agriculture management, because we can apply inoculation and help the association with mycorrhiza of the plant. Okay, going to the next one. Here are co-inoculation of these two bacteria, Azospirillum argentinense and Pseudomonas fluorescens, in this case for service crops or cover crops. If you can see in the picture that in some particular agricultural system, you need to cover the soil because the cash crop is not very good because it's not leaving good residues. So you can put this kind of crops like oats and rye in the soil and help to maintain soil organic matter. And if we use rye, as you can see in the bar figure on the left, could be increase the biomass of this crop with the inoculation of these two, the co-inoculation with these two PGPR bacteria. But the oats is not changing. So maybe we can have this opportunity to choose which crop we use in particular cases, helping with the inoculation of PGPR. I'm going to talk about some new, I said new because most of the literature is on Azospirillum, the Rhizobium legume associations, but recently we have a lot of information about this genus, the genus Bacillus. Bacillus inoculation have a lot of benefits for the plant. On the table there, you can see that the several species of Bacillus which can be used as a biocontrol for different pathogens, plant pathogens. And also, this list is not complete, it's just an example, because Bacillus have a lot of possibility to help the plant and promote its growth, like biocontrol of pests, biocontrol of bacterial disease, fungal disease, and also growth promotion as a direct mechanism. The important things to say also for this Bacillus PGPR bacteria is that they are very important in the rhizosphere. And if you make a program to select microorganisms, you always find good ones of this group. So in our lab, we make a program to select PGPR for biocontrol of Ralstonia solanacearum, which is the pathogen we produce the bacterial wilt in several crops, particularly in the Solanaceae family of plants, like tomato or potato. And we found a lot of, we have a collection of more than 400 isolates, and more than the 50% are Bacillus, and they have a good performance. Other thing which is important for this group is that they form endospores, so they are very good to formulate bioinputs because they have a very good stable formulation with this PGPR. Well, we are reaching the end, and I would like to say that the use of biotechnological application, like the use of microorganisms to formulate bioinputs, is a good strategy, but you have to think that we need to have in mind a lot of things to get good ones. And on the left and on the right, you have some kind of guide to go step by step to get good bioinputs from different agriculture systems. The first one, or the first step, should be choosing very good you have to choose very well where you choose the sample, where you take the samples. If you have a focus to control a particular disease, you have to check if you sample from ill plants, or if you take the soil, or anything you need, you have to sample, and after go to the lab and make all the procedures to confirm that you get isolate microorganisms, and after that you have to check if there are biological interactions, if the microorganism you isolate is going or is helping with the objective you have in the beginning. When you get that, you have to formulate. And if you formulate, you have to check that the formulation is stable. You can choose different formulation like liquid or solid and screen the alternatives. And after, with this information, you have to make greenhouse and field experiments to prove that the microorganism in the bioinput is helping the plant with the objective you propose. On the right, you can see also that if you decide you are going to use a particular formulation, you have to check different methods of inoculation. If you inoculate on the seeds, or you inoculate on the soil, or inoculate different times during the crop cycle, all these things should be established for a particular bioinput. And finally, you have to suggest or have to have the procedure how to control the quality of this bioinput. And the commercialization should be in a presentation that the farmer could use easily. So it's very important to check off this list, and the academy is always doing the first part, and the interaction between the academy and the companies is helping with the formulations and all the steps which are to get a commercial bioinput. In the center of the figure, you can see that the formulation can be having a different impact on the, by example, grain yield. In this case, I show you the results of a wheat field experiment with different formulations with the bacteria Azospirillum. And you can see there that two of them, the second and third bar, are increasing grain yield in the significant way. So the formulation is having the same bacteria, but the formulations are different. So there are other components in the formulation, and this component could have an impact in the quality of the bioinput. So I think I finished now. We can go to the last one. To finish with this dissertation, I would like to share with you these two phrases because they are some kind of abstract or summarize what all the ideas I try to show you here. Life on Earth is a symbiotic and cooperative union that allows those who join together to succeed. We can see this phrase stated by Lynn Margulis, a very significant researcher for the soil microbiology, that if we try to help the plant to produce more, we have to think in the symbiotic and cooperative unions with soil microorganisms. And also, if we have to success with bioinputs, I like this second phrase: the success of using bioinput is based on the selection of microorganisms, the selection of appropriate formulations, and the good inoculation design. Thank you very much.
We'll proceed now to the next section, which is the Q&A. So please keep your questions coming in. But in the meantime, I'll start with a question from Sushant. He asks about Azospirillum's role. And if it's not a legume crop, will it still help?
Yeah. Sometimes you can get a better growth and also a better nodulation if you co-inoculate rhizobia with other PGPR, like Azospirillum. So, and also rhizobia, which have the ability to produce phytoregulators, can help the plant different of legumes. this strategy are used sometimes.
Okay. Another question from Sushant about siderophore. How do you induce it? As well as when you talked about the indirect. So what about the phytohormone production, indoleacetic? Can you explain a bit more about those indirect functions?
I understood that the question is related to siderophores. Anyway, if
Yes.
Okay. Siderophores are particular substance that can chelate iron. So they transport iron from the soil to the plant. And the bacteria have the mechanism to produce, all the organisms produce siderophores, but the bacterial siderophores are more efficient. So if the plant is associated with the bacteria producing efficient siderophores, can increase the nutrition of iron. But because siderophores are molecules that can control other microorganisms like fungi, because the fungi siderophores are not very efficient. So this mechanism could help the plant with the interaction with bacterial siderophores to reduce the amount of some particular pathogen like a fungus.
Noted.
I don't know if I complete the idea or you need more explanation.
That should be okay. Our next question is about somebody who grows gypsophila in an open field, and whenever they get rain of more than 15 millimeters, plants die. They do trichoderma, bacillus, pseudomonas, a lot of biology, but still the plants die when it rains heavily. What can be done? They also have a second question. In commercial agriculture, how can we grow without fertilizer as we reduce fertilizer inputs? So two questions, one about how to grow without fertilizer. So reduce it to a point you probably use zero inputs. And number two, when you have, you think you're doing the biology right, but you still get challenges during heavy rains. How would you approach that?
Okay, going through the first question, you have to have in mind that the genotype of the plant was produced by the last century, the 28th century paradigm, the new revolution there. So this plant probably was produced with the necessity to add fertilizers, to add chemicals to control pathogens. So perhaps it's not prepared, the plant, the genotype is not prepared to receive the microorganism-plant associations. Because of that, I stress the importance of the plant genotype. Anyway, could be also another problems, like maybe the interaction between the biological supply is not the best, but in general, when the plant is in interaction with the biologicals, the plant is responding. But could be a lot of things. And this is connected with the other question because, as I try to explain, we are going, in this moment, we are going through two paradigms. So the paradigm of the use a lot of agrochemicals in the crops, and the new paradigm. And we need to change a lot of things to stay in the new paradigm. We are still going through. So that's, for me, is that the explanation.
Okay, Dr. Inés, thanks for that explanation. So Avinash had asked a question based on your last statement. 50 to 70 years ago, we were told that fertilizer and pesticides will improve our production. That is basically the old green model green revolution model that you talked about. In 2025, we are told that we have a new paradigm, which is called the new green paradigm, and it's supposed to solve the problems of the old one. What is the track record to prove it? And the education we have so far is based on NPK doses and pesticides. So can you compare what is working in the new one versus the old one, you know, and what have you used to make these experiments and, you know, to come to these conclusions that this paradigm is better?
Okay. One of the important things to know is the record that we have, that the humanity have, that the use of agrochemicals contaminates the soils and reduce the activity of soil microbial communities, reduce diversity. And diversity is a very important thing to take care of. And well, that's one way to analyze this. Also, if we use machinery associated with adding fertilizers and chemicals in the crops, we reduce, as humanity, the amount of soil organic matter. And the reduction of soil organic matter carry on a lot of problems. And if we change the amount of soil organic matter, we reduce soil health and soil quality. And the soils worldwide are responding to the agriculture management using different kind of management, like zero tillage, like cover of the soils, and also reduce the amount of chemicals. My conclusion is because I study this situation, I analyze this, and we say that the necessity to increase the information to go through the new paradigm. The bioeconomy paradigm assumes the agriculture sustainability and the industrialization with some idea which we call intelligent industrialization. We have to increase the amount of possibilities of the use of biological interactions. We have a lot of potential, and we need more applications. I hope I answered the question.
Yes, I think you did, and you answered his next question. He was asking what are the benefits of this new paradigm, and like you said, you definitely need to preserve diversity in the soil, and also remember that the machines and the equipment that you use to apply these fertilizers have some effect on the soil. So there's some people who are enthusiastic to adopt this new model and probably already have adopted it. So a question from Gift: how long will it take for soil to have a good biological balance after years of using pesticides and organic fertilizers and fossil fuels? So how long? If we start today, when can we expect changes? When do we know that our soil is good now?
Well, there are some information about that. It's not a very quick change, because the main thing we have to check is the diversity, but the diversity is associated with the amount of soil organic matter. And one parameter you can check is that soil organic matter, carbon, organic carbon in the soil, and the specialists say that should check this parameter at least every 2 years. So if we see changes in 2 years or 3 years, you are going in the good way. And there are informations that show that by example, the amount of soil organic matter was going down, and in one point, it's a kind of prediction using simulation models that if you change the agriculture management and stop the using of movement of soil with equipment different to make zero tillage, the soil organic matter began to change and increase. But it's a long movement. It's, as I told you, maybe every 3 years you see a change. But the change can be seen. And a lot of places in the world, they change the agriculture management to zero tillage, and the increase in soil organic matter had been reflected. So the soil organic matter change and the soils recuperate the amounts that they have maybe 60 years ago.
Perfect. Thank you for that answer. So for that question, it's a gradual process. So check every 2 to 3 years. With time, you should be able to see these changes. So I'm getting some concerns here from Rakesh. He's asking, so do these biologicals, these formulations, do they stay alive for long, especially when they're imported from different countries? There could be a lot of factors affecting the efficacy on transit, which the end user may not be able to know. So can you guide us on that from formulation to application? What factors should we take care of to ensure that we have the same efficacy that has been promised on the label?
Okay, the efficacy could change if the bioinput is not maintained in good condition, but the good conditions are different for different types of bioinput. It's not possible to make a generalization. By example, Bacillus is producing, as I mentioned, endospores. So this could be a more stable bioinput. But the formulation of different types of microorganisms are definitory to say how long is going to be a good bioinput. And the manufacturing should say that, but the farmer or the people in the middle should check that. The expiring date is a good parameter, but also the farmers, the manufacturers say different parameters that the bioinput had to have, and this is the only information we have sometimes. But if you know the bacteria or you know the type of bioinput, you have general indicators of quality control. By example, in the case of Rhizobia or in the case of Bacillus, you can see the expiring date should be more than that, all these kind of things and the amount of bacteria in the formulation should be regulated by different governmental part, no regulation. So it's not the same everywhere, but the quality control is very important. And the protocols to check that are very important. By example, in Argentina, we have in the Argentinian Association of Microbiology, we have a network working with different kind of microorganisms to get consensus protocols to check quality of the inoculants. And this is applied, and the companies and the farmers or the people in the middle can use these suggestions to evaluate the quality.
So, still on that question, are there like general factors that maybe probably apply across the board, like for example, temperature, pH? Can you advise on such?
Well, some manufacturer companies suggest that the bioinput stay with cold, but others, they said they don't need it. So it's not a general suggestion to make. Which is important sometimes is how the bioinput is applied in the farm, because it's very important to use a place with cover, no under the sun. The application, the inoculation of the seeds, by example, should be in a safe way. It's not recommended to use outside, and in some particular situation that could be provoking different problems. But in places with high temperatures, it's very important to use microorganisms which are adapted at this situation. But it's coming in the beginning of the process.
Thank you so much for that. So Rakesh, I hope your question is answered. There's no general one rule fits all, but look through the label, look at the advice from the importer, check from source, understand whether what you're getting is adapted for your climate. You touched a lot on mycorrhiza. So someone is asking, Kabilan, how long does it take for mycorrhiza to establish in the root system after inoculation? And also, number 2, can azospirillum, mycorrhiza, and trichoderma be applied together? So
Okay.
These are our final questions as we wrap up the session.
Okay. We check in different, as I mentioned, this is not inoculation with mycorrhiza. The information I show you is native mycorrhiza. But when we make the experiment in field conditions. So, by example, we make the sowing of wheat, for example, and when the plant is in tillering, maybe a month after of sowing, you can check the roots, take samples, go to the lab, and make the tinction of the roots and check the structures there. And you can find in that time several associations with mycorrhiza. So you find mycorrhiza after a month of after the sowing of wheat. If you go to the field anywhere and take a plant, the plant is always with mycorrhiza. The association in vitro experiment or maybe in arsenic conditions, where a particular mycorrhiza fungus is applied, show that the association is in a very short time, maybe less than a week. Respecting to the other question, if we think that the mycorrhiza in a very healthy soil is present, you can inoculate with Azospirillum, with Pseudomonas, and also with Trichoderma. There are also a lot of reports showing that, so it's possible.
Amazing. Thank you for that. So we have many questions coming in, but unfortunately, because of time, we won't be able to take all of them. But I'm sure the good doctor has given us a lot of insight from this session. So at this point, we will wrap up our meeting. I want to thank you all for attending. And to give the vote of thanks, I'll ask my colleague, John Ogecha.
Thank you. Thanks, everyone, for the opportunity to have you again on board on Root Camp episode 3. My sincere apologies for the rather difficult beginning, but I'm glad that we made it through. A special thanks goes to the organizing team, particularly CKC, our partners, and particularly Dr. Inés for that very insightful presentation. I'd not forget the CKC background team, particularly Lucia and the team that made this possible. We really appreciate, and I hope we should be able to do this more and more so that we educate our clients and partners. Special thanks also to the team that has been able to join this particular episode. We really appreciate it. And special thanks also to those who've been able to join all the 3 episodes. We are still going on with this. Since we believe that, always is the belief is that education comes before everything else. So we really appreciate your time for this.
We also encourage you, please join our WhatsApp. From there, you'll be able to connect with other growers. We'll be able to ask one-on-one questions, engage with our representatives. So please make sure you join our WhatsApp group so that we form a community of growers who are interested in sustainability.